Morena, everybody. Welcome to the morning session on the final day of the 2021 Forest Growers Research Conference. My name is Brent Guild, GM for 141 New Zealand, located here in the top of the south, and I'll be chairing today's first session, which is part of the industrial forestry and wood products theme, with a more specific focus on tree breeding technologies. We have 4 speakers lined up for you this morning, and the first of those is Craig Ford, who is a propagation research scientist at Scion Research. As well as pure propagation research, Craig's sphere of interest is in containerised production of pine seedlings, cuttings and tissue culture plantlets, as well as a keen interest in automation, mechanisation and disease management. Craig's presentation today is titled A Nursery Production System to Help Meet Our Changing Forestry Needs. He will be discussing 2 pieces of research undertaken at Scion recently, looking at opportunities to extend our radiata pine planting windows, in turn helping to meet the increased planting stock demands from our climate change aspirations, especially when issues such as labour shortages are impacting our industry. The aim of the presentation is to present evidence for new approaches to nursery propagation which will allow New Zealand to supplement its production of radiata pine outside of its normal bare root production and planting windows, while also responding to a changing operating environment. Next up, we will hear from Alec Foster, who will give us an update on genetic gain. Alec is Research and Innovation Manager for the Radiata Pine Breeding Company. He was taken on to bring a more commercial focus to RPBC's R&D activities, particularly the deployment of the Radiata Pine SNP chip developed as part of the Industry Genomics Research Program, and also to help identify research that brings value to the sector. Alec's presentation today will focus on how RPBC serves the industry by creating value through breeding and other activities, as well as the establishment and measurement of multiple trials in New Zealand and Australia, and the genetic gain that is being achieved. In addition, he'll share various new initiatives that are being explored, such as research into alternative species, climate change, endophytes, and software to further improve tree breeding and the distribution and deployment of genetic material. Thirdly, this morning, we will hear from Glenn Thorby on gene editing for forest productivity, resilience and sustainability. Glenn is originally from the UK and is a forest biotechnologist who has worked at Scion for 12 years. His research involves developing and using various biotech tools for tree improvement. In his presentation today, Glenn will describe why Scion is working on gene editing and why it is potentially a valuable tool for forest improvement. He'll then tell us how gene editing fits with other breeding technologies, then update us on progress with gene editing in radiata pine and Douglas fir, and finally he'll discuss research which Scion has carried out with the aim of developing sterile and low-coning trees as part of the MB Winning Against Wildlings research program, which received funding from FGR. Our final speaker in this session this morning is Loretta Garrett, also from Scion, who will talk about the contribution of long-term trials to the positive outlook for industrial use of forests. Loretta is a soil scientist who specialises in forest sustainability and carbon. Her research field is quite broad and encompasses the long-term sustainability of planted forests over multiple rotations, climate change impacts on soil and the delivery of ecosystem services, and the environmental fate of herbicides. In today's presentation, Loretta will talk to the matter of long-term forestry field trials being fundamental to our understanding and management of forest ecosystems. Trials provide the empirical evidence which help benchmark our understanding, allow us to observe trends. They're foundational to predictions and modelling, and are a place where new measurement technologies are developed. Her presentation today will highlight the value of long-term trials to the continued industrial use of forests. It will use the harvest residue removal trial series installed in the 1980s and 1990s as an example of how data-driven evidence can demonstrate continuous improvement in sustainable forest management, as well as highlighting how to get the most out of long-term trials and some of the challenges we face in keeping those trials going. So that's the run sheet for this first session this morning. The genetics and tree breeding arena can sometimes be quite dense in its subject matter, but I think that Bart and the team have assembled a really good range of speakers here who speak to a number of the practical challenges which the industry has been facing into for some time. Things like labour shortages, proof of gain, wilding management, and something that's dear to my own heart, and I know a lot of the foresters in the room, and that's the value of long-term trials to an industry that by its very nature operates in long-term planning horizons. So we look forward to that over the next hour. And just to reiterate, there is an opportunity to ask questions at the conclusion of the full presentations in just over an hour's time. And until then, please sit back and enjoy the time with our four speakers. Thank you very much.
Tēnā koutou. My name is Craig Ford. I am a plant propagation research scientist at Scion. Thank you to Forest Growers Research for the opportunity to present to you today. I'm presenting work completed by our Scion Research Nursery team. The aim of this talk is to present evidence for new approaches to nursery propagation, which will allow New Zealand to supplement our production of Pinus radiata outside of its normal bare root production and planting windows, while also responding to a changing operating environment. New Zealand has a proud history of rising to nursery challenges. During the mid-1990s, the forest nursery industry was able to quickly ramp up production of radiata pine to meet the high afforestation demands of the time, between 40 and 75,000 hectares per annum. This was almost exclusively produced as bare root crops, as shown in the picture, and also led to a flurry of research and publications. After an almost 20-year maintenance period, we are once again challenged with high afforestation demands. The Climate Change Commission recommends we establish an additional 22 to 26,000 hectares of exotic trees over the next 15 years, with an ever-increasing addition of native trees, shown in green on the graph, until we reach an approximately 50,000 hectares of new forest per annum between 2028 and 2032. How much of that green gap is taken up with natives versus exotics remains to be seen. The mid-2010s planting stock demand was easily accommodated by our national nursery infrastructure, then producing around 45 to 50 million trees per annum. By 2018 and 2019, this all started to change, with the introduction of the government's 1 billion tree program. Within a very short period, nurseries were required to ramp up radiata seedling and cutting production to around 90 million plants per annum for 2019 and 2020, and could exceed 100 million plants in 2021. The question is, how can we sustain this production and establishment rate with some of the industry challenges we face, especially if, as has happened to date, radiata is required to fill the gap in our afforestation goals, in the event that native trees aren't produced in sufficient number. While bare root is the predominant means of deploying radiata, there are a number of new pressures on this industry that weren't as prominent in the 1990s boom. Some of these pressures include increasing pest and disease threats, coupled with tightening chemical legislation and social license to operate. As these pressures increase, it is becoming more difficult to raise bare root crops. The picture on the right-hand side illustrates a crop produced in the USA without fumigation in the foreground and with fumigation in the background. The health differences are pronounced. While significant effort is being put into improving sustainability in this system, there is also a widening opportunity to introduce containerized stock, where containers are temperature or UV sterilized and grey media is not reused. One of the main limitations of bare root production systems is the high seasonality of the operation, with lifting, setting of new cuttings and field establishment all happening within a short 3 to 4 month period in winter. Finding labour to do these exhausting menial tasks over this cold and wet period is difficult. Changes in legislation and environmental pressures are driving the industry towards a more high-tech, precision-based nursery sector. where we can limit or eliminate the application of harsh chemicals, use integrated pest management approaches, reuse water and nutrients, and improve our biosecurity measures. While technologies like these do exist and can be adapted to bare root forestry, like the weeding robot pictured here, containerized nurseries are significantly more advanced in this area, and many of the required changes are already very well understood and developed throughout general horticulture. Using containerized planting stock provides further benefits when it comes to labor supply issues. By having a year-round production facility, containerized nurseries are able to offer more permanent positions for staff with improved working conditions. Containerized nursery stock also allows well-established mechanization and automation opportunities for the nursery and for outplanting, thus requiring less staff which are more skilled. To ensure that we can meet our afforestation aspirations with these limitations in mind, we need to improve and expand alternative nursery systems to supplement the current bare root capacity. Over the past few years, Scion has been investigating new systems to meet some of these needs. Firstly, we have implemented trials looking at a modern containerized forestry nursery system widely used in South America and South Africa, where we are able to raise, harvest and set radiata cuttings all year round. This is commonly called a mini hedge system. Secondly, we have revisited how containerized trees perform against bare root crops out of the normal establishment window in a simulated field establishment trial. Knowing that mechanized planting can be hard on containerized stock, we have also investigated the feasibility of new paper pot planting stock to address this potential issue. Hydroponic mini hedges were originally pioneered in the early 2000s in South America for clonal eucalyptus production, but now dominate the cutting sector in South America and South Africa, including for pine propagation. By enclosing hedge tunnels and providing optimal nutrition, you are able to maintain year-round production of highly juvenile shoots, which show excellent rooting success and good root architecture. The system also allows for the recapture of water and nutrients, which can be UV sterilized, filtered and reused. Pictured on the right are examples of mini hedge sandbeds in South Africa, producing Patula Techonomania hybrid crossings. Sappi Forests is currently deploying approximately 60 million containerized trees per annum from their nurseries, with almost half being rooted cuttings. Some South American nurseries individually deploy 50 million or more containerized cuttings per annum, almost all of which are in paper pot or are transitioning to paper pots. Pictured on the left-hand side here are trial mini hedges for radiata at Scion, which were established in 2020 and have performed exceptionally well thus far. In our radiata rooting trial, we set cuttings monthly for 8 months, outside of the normal bare root season, across 8 different genotypes. All cuttings were set into 4 centimeter Elipot paper pots of around about 125 cubic centimeters volume, which is the equivalent of a TS48 container. Results presented here are for a total of 52,000 cuttings which we set at Scion between August 2020 and March 2021, for which we have final rooting and dispatch counts. While hormone, growing media and paper type trials were also conducted, these are not reported here. For this report, we look at rooting across months and genotypes. Rooting was highly favorable across all planting months, where the minimum success was 84% in January and February and as high as 98% in November and December. Despite the fact that previous bare root research has shown a high degree of variability in rooting success between radiata genotypes, there was no significant differences between genotypes in this trial, where the lowest overall rooting was 88% and the highest was 96%. Our conclusions so far are thus that radiata is highly amenable to the hydroponic mini-hedge system of propagation, and year-round propagation success can be achieved even through summer. With learnings from radiata propagation technologies being implemented in South America, we chose to use paper pots, which performed well in the nursery and allowed for much easier lifting, packing and transport. Our second trial investigated the use of containers to extend our planting window. To test this, we established a field trial at Sion, where we planted bare root, standard containerized and various paper pot options at monthly intervals, starting in the beginning of the very dry and hot March 2019. To illustrate the opportunity of raising cuttings quicker and out of the normal season using containerized growing, all cuttings were placed in November 2018, meaning that at the first planting in March, most of the cuttings were just 4 months old from setting. The exception to this was the winter set bare root control, which was set in June 2018 and was hence 9 months old in March. The container types used in this trial to compare against the November and June set bare root were the Transplant Systems TS48 tray and the Hyco V150 tray with paper pot sleeves shown in the middle photograph, noting that we did not yet have the proper holders for these in the country. Paper pots were sourced from 3 different companies: Plant Paper, BCC and Elipot. The picture on the far left shows a 7-month-old cutting in a paper pot. The results presented here are a combined measure of the survival and growth of treatments at 2 years, measured as the total conical volume for the plot of 6 trees. As expected, survival and growth of the winter set bare root cuttings was highly seasonal, as illustrated by the red regression line on the graph. Despite being 5 months older than the other treatments, the June setting still performed poorly outside of the recommended planting window. In comparison, the November or spring set bare root cuttings performed poorly all round, especially outside of the June-July planting window. The TS48 plugs, however, did significantly better than all of the November bare root cuttings of the same age. The poorest performance of the TS48 container type was in March, where at just 4 months after setting, poorly consolidated plugs experienced quite severe transplant shock and survival was poor. The paper pots, represented here by the Elipot VP paper, also showed significantly better survival and growth compared to the November set bare root cuttings, but significantly outperformed both the bare root and the TS48 treatments when established in March. This suggests that the plug, being held together by the paper, has reduced the transplant shock and facilitated better establishment. This trial has reinforced our understanding of the bare root crop cycle and its seasonal limitations, but has also illustrated that, as has historically been suggested, containerized plants offer an opportunity to extend this planting window. This trial has also shown that there is an opportunity to successfully root cuttings outside of the normal production window and still have them ready to plant within about 6 months, with good establishment results. This has huge implications for an investment in a containerized facility, as double cropping reduces the required growing area and hence also the capital costs of such a nursery. While further work is required on refining paper pot recommendations, it certainly shows great promise in being able to improve survival of younger plants on harsh sites. Future work should extend our understanding of nursery and field mechanisation opportunities and requirements from plugs, and shorten our crop cycles in the nursery. A special thank you to the fantastic Scion Nursery team for growing, establishing and measuring these trials, especially to Bina Poi for the coordinating of the initial establishment and measuring, and Alex Lloyd for recent measuring and data collection. Special mention also to our science leader at the time, Heidi Dungy, for her support of this work at our nursery. Thank you. I welcome any questions in the question and answer section at the end of this session.
Good morning, everyone. My name is Alec Foster. It's a pleasure to be speaking to you all, even if it is only virtually. I work for the Radiata Pine Breeding Company, and I'm going to be giving you a presentation today, providing a bit of a background to the RPBC, the value that we create, and the exciting research that we're currently doing and planning on doing in future. So the RPBC supports the forestry sector by improving the genetics of the material that's planted. The better material planted, the better the commercial outcomes for forest growers. It's really no different to any other industry or sector. If you use better ingredients, better feedstocks, you're likely to end up with a better product, a better end outcome. So this is really what RPBC focuses upon. Now, the RPBC and its predecessor, the Radiata Pine Breeding Cooperative, have a long history of improving the germplasm within New Zealand, and as such, we own most of the IP. Our core business is breeding, as the name suggests, which in essence is targeted mating and characterising, measuring those traits of interest. So we have large trials across New Zealand, across multiple environments, helping separate what is the nature and nurture. Do we have a tall tree because it has superior genetics, or do we have a tall tree because of the environment that it's grown within? And these experiments and trials help us determine what are the best offspring and what are the best candidates for future mating. But we've also got to be conscious of many of the things that are being spoken about in this conference. Things like climate change, pests and disease, species diversity. And recently RPBC has updated its vision to include the risks and challenges as we move forward as an industry ahead. Now, RPBC as a company runs on a not-for-profit basis. Our income comes from both the shareholder levy and royalties, and this amounts to approximately $3 million per year. And this income goes straight into our breeding and research and development. We have nearly 200 trials across New Zealand and Australia with thousands of genotypes. And that is an immense amount of data, a wealth of information, helping us understand various trees under different environments. And most of this work wouldn't be possible without our shareholders. We've got 16 shareholders, and they represent approximately 60% of the Pioneers estate in New Zealand, 30% in Australia. Many of our trials are held on their estates, which provide us the invaluable information that we use for our breeding processes. Our team is only small. We've only got 6 employees. Mark, he's a tree improvement manager, so he's our breeder, so he's the brains behind a lot of our operations, selecting the best candidates, the best trees for distribution, the best trees for future breeding. Ben, he's the man in the field. He provides a lot of our on-the-ground work, helping establish the trials, helping us measure the trials. My role is associated with identifying new R&D, with commercial outcomes, helping understand how we can leverage a lot of our knowledge that's been built up over generations. I'm also have an important role working with Scion, helping operationalize the SNP chip and the genomics technology. We also have a fantastic support team. Belinda, Kelly and Kevin, and without them we won't be able to do much of the work that we do. Now, because we're only a small team, we need to rely on outsourcing most of our work, and it'd be impossible to work without our great collaborators. We have a close relationship with Scion, trying to explore the next generation of R&D and blue sky research that needs to be performed. We have multiple teams helping us utilize the genetics, companies which help in terms of our general day-to-day operations and working in the field, consultants with international expertise, and we've also got 2 great PhD students at the University of Canterbury. Now, RPBC's breeding objective is really the traits that we try and prioritise. So this is our relative weighting to some of the most important aspects. So RPBC prioritises volume because this has the greatest impact on value for the industry. But we're also very conscious of wood quality traits such as density and modulus of elasticity. And we measure trees at approximately 10 meters height, which is an 8 to 10 year kind of age range. And this gives us a good prediction of what's going to be the characteristics the trait is, the tree is at harvest age. Now, the main thing that most of the industry wonders is what is the value of RPBC and the germplasm that we provide. I really quite like this graph. It gives an indication of the distribution of the increase in value per hectare. So this is a distribution from 1997 onwards, so over 76,000 measurements. RPBC is continuing trying to push this distribution curve to the right, and in 2020 those included in our crossing program had an MPV per hectare increase of $2,218. So that gives you an indication of the value of the genetics that RPBC is generating, and also the historic value that has been generated over time. However, moving forward, RPBC recognises that we need to ensure the uptake of our material, ensure that our research is translated into actual commercial outcomes. So we're undertaking a number of initiatives over this next year or two to help make this a reality. The first thing is we're going to be engaging the industry far more face-to-face interaction. We're going to be doing research about understanding what the industry requires and needs. This will help us inform our breeding objective and tailor it better to the needs for 2030, 2040, 2050, thinking about the future and how this changes. We're also developing a new support tool to help germplasm deployment, so helping provide transparency to our different seed lots and the various benefits or negatives associated with each. We're also going to be improving our communication. One of these ways is translating from our GF system to EBVs, and this will help articulate the value that we bring. The last thing, and really quite important, is we're going to be introducing a second breeding orchard. This second breeding orchard will help increase the breeding intensity, and it will also provide a mechanism for us to use genetic selection using our SNP chip. We've also got to realise the value from our SNP chip. Now, I'm sure over the last 8 years, you've all heard about the SNP chip that's been developed between RPBC and Scion, and it's fantastic. We have done great work developing this R&D tool. However, the next stage is actually translating it into application, realising the value from the SNP chip. So this involves RPBC developing a robust pipeline automating the process and bringing down the cost, making sure that we can actually use it on a day-to-day basis for genomic selection. And next year, RPBC are planning to use genomic selection for the first time. And this will cut down our breeding cycle from approximately 17 years closer to 10. It's also going to be useful for pedigree verification, identifying at a genome level, what tree we actually have, rather than what we believe we have. Once we've got this, there's also an immense amount of work that needs to be done, and we're starting to initiate this work with science today in terms of understanding genome-wide association studies. So at the moment, the SNP chip and the SNP data is effectively a black box. We want to translate that into knowledge, and this knowledge will help inform genetic engineering and also the genotype by environment. Now, the hot topic moving forward is climate change. RPBC simply can't be performing breeding in isolation of the changes in the environment in terms of climate change, in terms of pests and disease. And RPBC is increasingly aware of the need to adapt. We're increasing our germplasm diversity, introducing hybrids, other softwoods, working with Tree Breeding Australia. We've got a PhD student just starting at the University of Canterbury looking at red needlecast, and we've also been working with Jenny Aitken identifying endophytes associated with Pinus radiata. All of these, we hope, will mitigate a lot of the risks that we see on the horizon. I'd like to acknowledge multiple people, and there's too many to name them individually. I'd like to thank FGR for arranging this conference and for much of the work that they do in this area. There's considerable overlap with what FGR do and what RPBC needs to be looking at from a breeding perspective. Mark and Ben, these are the guys who are responsible for generating and measuring a lot of the material that goes out into the sector. So I'd like to really thank those guys. Our research partners and contractors, again, far too many to mention, but I'd like to single out Scion. Our close relationship is really important for RPBC and the next generation of research that we need to be performing. I'd also like to thank our shareholders and royalty payers. It is the income that we receive from this which helps us do the research that we perform. So thank you, everyone. Thank you. Bye-bye.
So good morning, my name's Glenn Thulby and I'm a research scientist at Scion working in projects involving tree biotechnology. So as you can see from the title, today I'm going to talk a little bit about gene editing and its potential uses in forest productivity, resilience and sustainability. And to start with, I want to really put this into a little bit of context about why we're doing this work. And central to this is the information that you can see in this slide. And this is really that mutations, or the rare errors that occur during DNA replication, are what leads to genetic variability. And so ancestors which happened in either you or your ancestors define what you are. And these mutations are really the basis of natural selection. And in this slide you can see a collection of birds with different shaped and coloured beaks. And these beaks have occurred by natural selection, and it's actually been a whole series of different mutations that have accumulated over a long time period to give this degree of variance. But this can happen over a shorter term period, and you can see that by looking at the moths on the left of the screen, and if you can make out one of these moths is black and the other's white. And the black moth developed with a single mutation which occurred during the Industrial Revolution in Britain when pollution was making everything very black and camouflage was better if you were black. And of course these mutations are also the basis for artificial selection or breeding. And rather like the birds, some of these have taken a long time and involved the build-up of a lot of mutations. This is the case with corn, which you can see in the little picture here, which was first domesticated about 10,000 years ago. And the corn we see today doesn't really bear any resemblance to its ancestor. And that's because a whole series of different mutations have been selected over a very long time period. But there are also examples of domestication where the changes haven't been so profound and so many mutations haven't been needed. And brassicas that we eat are a good example of this. And all of the examples you can see in this other part of the slide are actually the same species, and they have relatively few mutational differences between them. But these give different structures which have led to different crops developing. By about the 1950s, it was become apparent in a lot of crop breeding programs that there wasn't actually enough variability available to develop the crops further, which was what was needed in order to get higher yields to feed the growing population. So methods were needed to generate more variation. And a method that you can use to do this is called mutagenesis, and this leads to mutation breeding. A mutagenesis is simply a technology which allows you to increase the rate of mutagenesis. So going from that low background level to higher levels so that you can actually have more variation. And there are several ways you can do this. You can do it with either chemical or physical mutagens, and a lot of different methods have been tried. On this slide I've put an example of one of the more interesting ones, which is radiation breeding. And this is what's called a gamma garden, and these used to be very common, but now there are very few left. This one still works in Japan, and at the centre of this garden is a radiation source which emits gamma rays. And in order to carry out mutagenesis, plants are planted in this garden. Near to the source they get a lot of radiation, further away they get less radiation. And after a period of growth, these are taken away and then examined to see if they've got any interesting phenotypes. But the problem with mutagenesis breeding is that it's a random process. Mutations are generated at random right across the genome. And so you have to mutagenize a very large population and screen it to find interesting mutations. And even if you find one, the chances are that this mutation will be accompanied by undesirable mutations in the genome. So you have to do a lot of crosses in order to sort out what you want from what you don't want. This makes this fairly unuseful in conifers, shall we say, because of the long life cycle, we don't have the chance to do all of these crosses to purify things, so it's not practical to use it. However, it has been very useful, and over 3,000 mutant varieties have been developed, and many of these are actually in the foods we eat every day. And I guess this is where gene editing fits in, because gene editing is really a way of creating designer variation. We're actually changing a gene that's already in the genome, so it's a form of mutation. And in fact, the mutations you get are of exactly the same type if you get natural mutations or if you use these mutagenesis protocols, and so you can't distinguish them. This has led to many parts of the world regulating this sort of gene editing as a non-GMO, although in New Zealand it is currently regulated as a GMO. But the real advantage of this technology is that it allows you to make highly precise targeted changes. You don't have this random process. You can actually go into the genome and change a particular gene. So it's knowledge-based, and you can go in, change the gene you want, and hopefully then get the phenotype you want. And this slide just shows you how this process actually works. And I'm talking here about CRISPR, which is the gene editing technology that we use at Scion, but there are a number of gene technologies you can use. This is a very fast-moving field, and new ones are developing all the time. But in the case of CRISPR, the mutagen, if you want to call it that, is actually an engineered nuclease, something that we develop. And it comprises of two different elements. In orange, the DNA targeting part, and then another part, the nuclease, which is really something that cuts DNA. And you design the targeting part to a particular sequence of DNA, and this directs the nuclease to exactly that place in the genome. And when it arrives, the nuclease cuts the genome at the precise point you want, and that's the first stage of making a mutation. And so next I wanted to just mention some of the advantages of using gene editing in forest trees. And by forest trees, I'm particularly talking about conifers grown in New Zealand's planted forests. And I guess the biggest advantage is that forest trees are undomesticated. They've had very few rounds of breeding. As you can see, 3 to 4 in most cases. This compares to the thousands of rounds in things like corn. And because they've only had these few rounds of breeding, there hasn't been an opportunity to bring together all these desirable mutations to get phenotypes we would like. In fact, we don't even have the collection of mutations we would like. And so gene editing allows us to overcome this in a very speedy and rapid way. Because if we have a gene where we know that we would get a good phenotype by changing that gene, we can actually go and do it in one round. We don't change the rest of the tree, and so we can very rapidly introduce new desirable changes. And there are many applications that we can use gene editing for. And if you follow the popular press, there's these days often a story about a new gene-edited crop which is about to be tested or released. And many of the same things can be done in forestry. And key traits that we could improve are things like forest health, where we can gene edit to give greater disease resistance. We can also improve productivity and sustainability. And for the majority of the remainder of the talk, I'm going to talk a little about a sustainability trait, particularly dealing with the wilding problem in New Zealand. And that's because we've just finished a large program looking at this, and it was partially funded by the forest growers. But before I get to that, I wanted to say a little bit about the technology we've actually developed at Scion and how we're using it. And as you can see on this slide, we've actually developed editing and edited several genes in both Pinus radiata and in Douglas fir. And as I'm going to say in a moment, we've edited Douglas fir for sterility genes as part of the Winning Against Wildings project. But we've also done a lot of editing in Pinus radiata, and as it says on this slide, we've edited several genes which are related to the structure of cell walls, and we've also edited some genes related to sterility. And you can see a picture here of some Pinus radiata trees which actually have edits in a cell wall gene, which we're growing on a glasshouse to see if the phenotype's what we expect. But for the range of talk, I just want to say a bit about the Winning Against Wildings project. And as I said, this has been a 5-year programme which has recently finished, which was partially funded by the forest growers. And this project really encompassed all aspects of the wilding problem in New Zealand, and there was a focus both on the existing wilding problem and on preventing future wildings. And it was in this prevention aim that we worked. And our focus was on Douglas fir, and the reasons for this are that Douglas fir is a commercially important species. It's planted commercially, unlike many of the trees which create wildlings, and it's also very wilding prone. And so if we're to continue to be able to plant Douglas fir, we need strategies to make sure it doesn't generate future wildings. And we took 2 basic approaches to do this, which I'm going to talk about today. The first of these was the propagation of naturally low coning lines. And in Onslow 1 seed orchard, they had some trees which they'd identified as being low coning. Year on year, they produced less cones than the other Douglas fir trees in the orchard. And we wanted to propagate these trees, see if they preserved their low coning nature, and test them out in the environment. And the second thing we wanted to do was use the gene editing technology I've just spoken about to see if we could use this to develop some sterile trees which couldn't generate wildings. And so at the start of the project, we had to develop some technology to enable us to do it. And the first technology we had to develop was some tissue culture capability, because at the start of the project, we didn't have a plant regeneration system for Douglas fir. And we successfully did that in the first couple of years. And you can see along the bottom of this slide what we developed, which is a cell culture system, allowing us to go from some cell cultures through somatic embryogenesis through to some trees, which we can have out in the glasshouse and ultimately the field. We also needed to identify a set of genes that are essential for reproductive development. And that's because our strategy was to gene edit these genes so that they were no longer working. And if you took out an essential gene, they wouldn't be able to reproduce, and you would have sterile trees that couldn't generate wildings. And before I summarise what we've done with gene editing, I just want to say that the low coning strategy has been successful. We've managed to propagate these trees. They're about to go to Scion's nursery for some further growth before they're field tested by Onslow 1 in some different environments to make sure that in each environment they're still low coning. A challenge with this is it's a long experiment. We have to wait for them to reproduce. And really my final slide is just to tell you where we've got with the gene editing. And as you can see here, we've successfully edited 3 different genes we believe are essential for reproduction. And the first of these genes, or the first one which we edited, DAL21, now has trees ready to go into the glasshouse. And you can see some little pictures of these trees on this slide. And we're going to plant them in the glasshouse so we can check that they grow normally. Because although we think we've edited reproduction, we want to make sure we haven't affected anything else and that the trees grow normally apart from being affected in reproduction. We have these lines stored in cryopreservation so that we're ready to use them in future if we need to take them out again. The big test that we have in this project is how we're going to test sterility. Because, like the low coning trees, it'll take a long time, and currently in New Zealand there isn't a regulatory route to allow us to do this. We can't grow these trees outside because they're classed as GMOs. So we can't grow them to the stage where they reproduce and test whether the reproduction works. So we're a bit of an end here. We have to really wait for the government to change its legislative stance or look at doing this overseas. And to finish, I would just like to acknowledge the people who have actually done this work rather than me talking about it. And these are listed on the slide. Charleston and Laurel have done a lot of the CRISPR design and development. Steffi has done a lot of the screening, and Barbara and Kathy have been instrumental in developing the tissue culture technologies.
Tēnā koutou. My name is Loretta Garrett and I'm a soil scientist at Scion. I have been a researcher for 20 years in the area of forest sustainability and carbon. Today, I'll present on contribution of long-term trials to the positive outlook for industrial use of forests. New Zealand planted forests are a significant contributor to New Zealand's economy. They provide timber and fibre for industrial use in New Zealand and overseas. Our forests also support the New Zealand products industry, our sawmills and processing plants. The contribution of our planted forests is even more emphasised when compared to other land uses. For example, far exceeding sheep and beef farming when compared on a per 1,000 hectare basis. At a national scale, our planted forest timber investment is valued at $60 billion. Planted forests also deliver numerous ecosystem services that benefit New Zealanders. For example, carbon capture, erosion control, and regulation of hydrological processes, which further adds to the value of our planted forests. We also have the lowest fertiliser input when compared to other land uses in New Zealand. This puts forestry in a positive position as a low nutrient input land use industry. We do have a number of pressures to plan for. One is the demand of wood and fibre from production forests is increasing as numerous countries seek to transition to circular bioeconomies. This includes the use of harvest residues for bioenergy. To meet this demand, more is being asked of each hectare of planted forest land. There is a drive to push for higher productivity per hectare through improved genetics, higher stocking and shortening forest rotations. Meeting this demand with our current planted forest land area will place greater demands on forest soil resources. We also have uncertainty with our changing climate. Some impacts will be positive, while others are likely to be negative. And there are many uncertainties. For example, the impact of soil nutrient cycling and delivery of nutrients for plant growth. Forest productivity is underpinned by the long-term productive capacity of the soil. Site productivity can be represented by stacking productivity gains, which includes understanding the site and soil. Retaining the productive capacity of the soil is key, as once reduced, it is very difficult to get back. Over 3 decades ago, this issue prompted significant investment in long-term research trial infrastructure to gather evidence for how to manage forest soil nutrients sustainably. We are supporting a $60 billion investment in forestry, so how do long-term trials de-risk our investment? I will now like to step through one example of a positive impact for the industrial use of forests from a long-term trial focusing on nutrient supply. The long-term trial has gathered evidence on impacts of harvest biomass removal from stem-only extraction through to a very extreme experimental treatment which removes the whole tree plus the forest floor. New Zealand has a set of these trials that have reached the end of rotation and which are connected globally to a network of harvest removal trials. We are able to demonstrate with data that all sites are not equal for the total amount of nutrients on site. The soils that support New Zealand's planted forests range from resource poor through to fertile. Where soil nutrients are low, the forest floor and harvest residues are an important store of nutrients on site. For example, at Woodhill, the forest floor made up 21% of the nitrogen on site, whereas at Golden Downs it only made up 2% of the nitrogen on site. We were able to demonstrate with data the percent of nutrient removed from the ecosystem varied by site and by harvest method, and prove that what biomass is removed from what site matters. For example, at sites like Woodhill that have low soil nutrients, a greater percent of the forest floor nutrients are exported with the biomass harvesting when compared to other sites. And higher nutrient sites will be less vulnerable as the soil holds the majority of the nutrients in the forests and is able to buffer the nutrient export with the biomass removal. We were able to demonstrate with data that there were low impacts on site nutrient supply and tree productivity with the removal of biomass, and only at low nutrient sites was the long-term nutrient sustainability of the soil negatively impacted with the removal of the extreme treatment of whole tree plus forest floor. The impact on tree productivity was also limited to low nutrient sites where the forest floor had been removed, and there was no impact on wood quality with the removal of increasing biomass. Therefore, the conclusion was site-specific management matters. The results of the harvest removal trials provide confidence that the productive capacity of most sites won't be impacted on after one rotation. We are able to de-risk our $60 billion forestry investment through including site-specific nutrient management. This confidence extends to regional economic growth and the supply of timber, carbon storage and reporting, nutrient balance model for nutrient tracking, and forest companies developing site-specific nutrient management plans. We need to celebrate these positive impacts and start influencing the public and government with our data-driven good news story. Doing this will deliver the full value of undertaking long-term trials. We have the evidence to demonstrate that the industrial use of forests can be done wisely and responsibly with site-specific management for environmental sustainability. This evidence is important as the environmental, social and ethical criteria will increasingly determine investment and consumer preference, and these long-term trials are supporting us on this journey to meet the required criteria. I would now like to reach out to the wider long-term trial infrastructure and how to get the most out of them. This diagram shows a selection of trials that Scion has on a timeline. The black horizontal line represents this year. Sitting behind these trials are many permanent sample plots that have many uses across a range of projects. What you will notice is that there are a number of trials that have passed the end of their rotation and now gone. The graph at the bottom shows the number of registered trials at Scion. You will see a peak trial installation time was from the 1980 through to 1990. We are now at the end of this rotation for peak trial installation time. We have been very lucky to capture a number of long-term trials at the end of their rotation. The harvest removal trials are one example. We have Puraki Experimental Forest that offers a 50-year legacy. We have trials that were installed under the Growing Confidence in Forestry's Future program that are the start of the next legacy. And we will have new trials going forward to deliver to future research needs. We are installing fewer trials than we have in the past, making what trial infrastructure we have more valuable. There is also a need to move towards experimental forests, moving away from small plots. to allow for forest level integrated landscape research over multiple rotations. Puraki Experimental Forest is an example of this, with 50 years of research with national impact in water quality, forest productivity through to carbon stock modelling. The trials we have now are capable of high impact. This can be achieved through targeting trials for data collection that will address multiple research areas, from improving uniformity, productivity and stand dynamics, understanding the role of the microbiome in forest resilience, through to improved phenotyping and modelling tools. This will deliver evidence for environmentally sustainable and financially viable forest management. To gain the confidence that our industrial use of forestry can be done wisely and responsibly, we will need to demonstrate to the public and government our continuous improvement in forestry management to gain the full value from these long-term trials. There are some challenges in keeping trials going and getting the basic data out. Some challenges are beyond our control, while others we could look to influence going forward. I would be keen to hear some of your suggestions on this area. If we take the harvest removal trials as an example, we lost one site in Golden Downs to an extreme wind event at mid-rotation. 2/3 of the trial area was flattened. We lost 2 trials to land conversion at mid-rotation. They are now a dairy farm. With 3 decades of time, we have had 3 generations of scientists and the same, if not more, forest managers. During this change, it can be difficult to keep consistency and maintain trial security. We have had an uncertain ride with funding, even to do the basics of measuring a tree, with patches of 3 to 6 year contestable funding projects. With the last contestable funding project coming through the Growing Confidence in Forestry's Future program, coming in just in time to collect the end of rotation data from 3 of the sites. We have seen how long-term trials can create impact for New Zealand forestry. The next generation of trials will continue to refine our knowledge as we move towards an uncertain future. To do this, we need to protect what we have and making the long-term trials work hard by delivering to multiple research areas. And to gain the full value, we will need to engage with the public and government to share and demonstrate that the industrial use of forests can be done wisely and responsibly. This is how you create impact. I would like to acknowledge the funding to capture end of rotation data from the harvest removal trials, which came from the Growing Confidence in Forestry's Future program. Funding to deliver this presentation from the Resilient Forest program, and a very special thanks to the many people over the decades who have contributed towards the harvest removal trials. Ngā mihi. Thank you for listening. I look forward to your questions on the contribution of long-term trials to the positive outlook for the industrial use of forests, and your suggestions around how to address some of the challenges we face with keeping long-term trials secure going forward.